Assalamu alaikum. In part 2, we want to continue talking about regulation of glucose metabolism and energy production. Now, it's known that normal blood glucose concentration ranges from 65 to 100 milligram per deciliter. We have a condition called hypoglycemia, causes weakness, sweating, and rapid heartbeat, and in severe cases, low glucose in brain cells causes mental confusion, convulsions, coma, and eventually death. However, in hyperglycemia causes increased urine flow as the normal osmolality balance of fluids within the kidney is disturbed. Prolonged hyperglycemia can cause low blood pressure, coma, and death. Now, insulin is released when blood glucose concentration rises. Its role is to accelerate the uptake of glucose by cells. On the other hand, glucagon hormone is released when blood glucose concentration drops. Glucagon stimulates the breakdown of glycogen in the liver and the release of glucose. So here we have this scale, normal fasting from 65 to 110, and hypoglycemia below that, and hyperglycemia above this. Then 140 fasting level diagnostic for diabetes, 140. 180 renal threshold, approximately level at which glucose appears in urine, at 180. Now, in the absence of food, a gradual decline in blood glucose concentration is accompanied by an increased release of glucose from glycogen. As glucose and glycogen reserves are exhausted, metabolism turns to breakdown of proteins. Protein is used at a rate as high as 75 grams per day. Lipid catabolism is mobilized and acetyl-CoA molecules derived from breakdown of lipids accumulate. Then acetyl-CoA is converted to ketone bodies. The brain and other tissue can switch over to producing up to 50% of their ATP from catabolism of ketone bodies instead of glucose. By the 40th day of starvation, metabolism has stabilized at the use of about 25 grams of protein and 180 grams of fat each day. With adequate water, an average person can survive in this state for several months. Those with more fat can survive longer. Now, metabolism in diabetes mellitus is one of the most common metabolic diseases, type 1 or juvenile, and type 2 or adult. Recently identified is a pre-diabetic condition called metabolic syndrome. Now, the symptoms of diabetes type 1 are all these. You can read them. Don't worry about them. Type 2, drugs that increase insulin or insulin receptor levels are effective. Metabolic syndrome has elevated. It's characterized by abdominal obesity, elevated. Again, don't worry about this. Autoimmune disease. Now, to treat type 1 diabetes, the missing insulin must be supplied by injection. This is the important point. Individuals with diabetes are subject to cataracts, blood vessel lesions, and gangrene in the legs. Now, ketoacidosis results from the buildup of acidic ketones in the late stages of uncontrolled diabetes. Now, this condition can lead to coma and diminished brain function, but can be reversed by timely insulin administration. Hypoglycemia or insulin shock, by contrast, may be due to an overdose of insulin or failure to eat. If untreated, diabetic hypoglycemia can cause nerve damage or death. Diagnosis and monitoring of diabetes, for those who are concerned. Random blood glucose concentration without fasting greater than 200, fasting blood glucose greater than 140, sustained blood glucose greater than daily. The blood test is desirable because it's more. Then this is not required from you per se. This is for interested people. Now we come to glycogen metabolism, glycogenesis and glycogenolysis. Genesis means synthesis and lysis means breakdown. Then glycogen synthesis, known as glycogenesis, occurs when glucose concentrations are high. It begins with glucose 6-phosphate. And you can follow these here. Relative change versus hour of starvation. Blood glucose drops and then liver glycogen start breaking down. Then we have fatty acid, etc. Glucose 6-phosphate is isomerized to glucose 1-phosphate by phosphoglucomutase. Now this glucose residue is then attached to uridine diphosphate, UDP. Now the diphosphate is then hydrolyzed, yielding to hydrogen phosphate ions. The resulting glucose UDP transfers glucose to a growing glycogen chain in an exergonic reaction catalyzed by glycogen synthase. Now, glycogenolysis occurs in 2 steps. Glucose 1-phosphate is formed by the action of a glycogen phosphorylase on a terminal glucose residue in glycogen. Then this glucose 1-phosphate is converted to glucose 6-phosphate by this mutase. Now, in muscle cells, glycogenolysis occurs when there is an immediate need for energy. Because glucose 6-phosphate cannot cross cell membranes, liver cells contain glucose 6-phosphatase, an enzyme that hydrolyzes glucose 6-phosphate to free glucose. Then, know this, that the liver contains this enzyme called glucose 6-phosphatase, which hydrolyzes glucose 6-phosphate to provide free glucose to go to the blood. Biochemistry of running, read it by yourself. Now, gluconeogenesis, glucose from non-carbohydrate. Now, lactate is a product of glycolysis in red blood cells and in muscle cells during vigorous muscle activity. Lactate absorbed from the blood is converted to pyruvate, the reactant for the first step of gluconeogenesis. Now, the new glucose synthesized in the liver is then returned to the muscles. This is called the Curie cycle, is named for Carl and Gertie Curie, who won the Nobel Prize in Medicine in 1947 for their work on glycogen metabolism. Again, gluconeogenesis, synthesis of glucose from non-carbohydrate precursors such as lactate and pyruvate, glycerol and the amino acid alanine. And there is the so-called the Curie cycle. This Curie cycle operates between liver and muscle. Now, gluconeogenesis begins in mitochondria with conversion of pyruvate to phosphoenol pyruvate. Pyruvate formed from lactate is transported from the cytosol into the mitochondria or is produced there from amino acids. In step 1 of gluconeogenesis, pyruvate is converted to oxaloacetate by pyruvate carboxylase in an energy-expensive reaction that bypasses the last irreversible step in glycolysis. Next, oxaloacetate is reduced to malate, transported into the cytosol and reconverted to oxaloacetate. In step 2, the phosphoenol pyruvate carboxykinase adds a phosphate group and rearranges oxaloacetate to produce phosphoenol pyruvate. This is the first of 3 instances that bypass irreversible steps in glycolysis. Because gluconeogenesis is not the exact reversal of glycolysis, because we must bypass the 3 irreversible steps of glycolysis, step 1, 2, 3, 1, 3, 10. Now, this is we bypass reaction number 10. The next 5 reactions of the 10-step glycolysis are reversible. They occur in reverse, catalyzed by the same enzymes. Now, step 8 is the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. And we have this enzyme called fructose 1,6-bisphosphatase, removes a phosphate group by hydrolysis in an energetically favorable reaction. So here step 3 is reversed. Then the next to last reaction converts of glucose 6-phosphate to glucose 6-phosphate, fructose to glucose. The final reaction in the pathway is the third bypasses The third, bypass of a glycolysis pathway reaction. Glucose 6-phosphatase now hydrolyzes glucose 6-phosphate, producing glucose and inorganic phosphate. Now, the glucose produced crosses the cell membrane and is transported to cells that use only glucose for energy generation. This pathway is important for cells and tissue that cannot metabolize except glucose, like مثلا the brain, red blood cells, cornea and lens of the eye, etc. So these tissue cannot metabolize except glucose. Now glycerol is also a precursor for gluconeogenesis. Glycerol is converted to dihydroxyacetone phosphate and enters the gluconeogenesis pathway at step 7. Now the carbon atoms from certain amino acids enter gluconeogenesis as either pyruvate or oxaloacetate. From certain amino acids, yes. For example, alanine. Alanine can enter here. Now the pathway begins at the bottom. So from top to bottom this is glycolysis, and from bottom to top it's gluconeogenesis. So the pathway begins at the bottom and moves upward. Enzymes shaded in blue are involved only in gluconeogenesis but not in glycolysis. For the other steps, لاحظوا, gluconeogenesis uses the same enzymes as those used in glycolysis. So here we have 4 different enzymes that are used in gluconeogenesis: pyruvate carboxylase, phosphoenol pyruvate carboxykinase, to reverse step 10, and to reverse step number 3 in glycolysis we use fructose 1,6-bisphosphatase, and to reverse step 1 of glycolysis, but it is step 10 for gluconeogenesis, we need the enzyme called glucose 6-phosphatase. So how many ATP and GTP are required? Remember, to synthesize 1 glucose you need to start with 2 pyruvate. So you multiply these by 2 to 2. So you have here, you must use 2 ATP here plus 2 GTP plus another 2 ATP. So a total of 4 ATP plus 2 GTP are required for the synthesis of 1 molecule of glucose. Chapter summary. This summary, after you read the PowerPoints or after you listen to this record, you can read these by yourself. These are summary of what we talked about in this chapter. And in this, for example, number 7 here, in number 7, what is the role of gluconeogenesis in metabolism? It maintains glucose levels by synthesizing new glucose from lactate, from certain amino acids like alanine, and from glycerol derived from fatty acid. This pathway found in liver cells only is part of normal metabolism and is critical during fasting and starvation. The gluconeogenesis pathway uses alternate enzymes for the reverse of the 3 highly exergonic steps of glycolysis, but otherwise utilizes the same enzymes for reaction that run in the reverse of their direction in glycolysis. We mentioned 4 new enzymes that are used in gluconeogenesis but not in glycolysis. The one in blue in the last figure we saw. So this is end of part 2, and thank you.